I would like to thank the organizers for inviting me to be here today. It's been a very interesting roundtable. And what I'm going to talk to you today is some of the work that we've been doing on liver organoids. Since I established them when I was a postdoc in Hans' lab, which he mentioned earlier before, I had put that I was going to talk about regeneration. There was a lot of questions on regeneration, actually, but I don't have time to go into that. And since I'm on the disease modeling session, I'm going to focus on that part. And just to mention, I'm moving my lab to the Max Planck in Dresden at the end of the year. So just briefly, the liver is made of many cell types. It's a very complex organ, but the epithelial cell types, which are the ones that are going to do the function of detoxification and metabolization, are the hepatocytes, and they do not work alone. They work really in conjunction with the other cell types in the liver. So indeed, if we want to make a proper liver organoid, we should try to aim to combine the different populations. This is not what I'm going to talk today. In my lab, we're trying some of these approaches, but I don't have time to talk about that. What I'm going to talk today is about adult liver organoids that I developed when I was a postdoc in Hans' lab, and how we've exploited them to understand disease mechanisms. I just put that slide, which I think it fits with what was the last discussion, to try to kind of generalize. The liver organoid word has been used for liver bat and also cholangiocyte organoids derived from iPS cells that came from Takeva's lab and also from Ludovic's lab. It has also been used by us to define these three-dimensional structures that we are going to see in a second, that they derive from, now we know that they derive from ductal cells in the liver, and these we call them now cholangiocyte-derived organoids. At the beginning, we just called them liver organoids, and we realized after Hans' paper and Nuss's paper that we had to call them cholangiocyte-derived organoids. Hans already mentioned about the hepatocyte-derived organoids. And I want to show here that actually the new manuscript from Hans in Cell and also from my lab in development, we have shown, and I will show one slide on that, how we can take this very early hepatoblast, which is a bipotential cell, and generate both hepatocyte and cholangiocyte organoids from this bipotential cell. So starting with the adult organoids first, this protocol, we established it in 2013 when I was, as I said, still a postdoc in Hans' lab. And what we knew at that moment is that we can take a healthy liver tissue, dissociated it. We didn't exactly know which was the cell of origin at that moment, which it came up before as a question. What is the cell of origin? In 2013, we didn't know yet. We knew it had to be something around the ductal tree, but we didn't know it was a ductal cell, maybe a portal cell around. And when we put it in the combined media, that it was first EGF and a responding, like the intestine, but no noggin, no BMP inhibition. And we add on top of it the famous nicotinamide and HGF and FGF. Indeed, these cells cell organize, and you can even start with one cell, and they will generate an organoid structure. Now we know that the cell of origin is a differentiated ductal population. This is not a rare stem cell. It's not a rare cell in the liver. Actually, the efficiency is extremely high to be a rare cell, so it couldn't be. So it's indeed a differentiated population that in vitro, it differentiates into a bipotent state, starts proliferating, and it takes 40 hours for the cell indeed to start S phase to proliferate. And it is capable still to generate now two different cell types, hepatocytes and or ductal cell, depending on which media you use afterwards to differentiate them. So actually, these cells in that state, in this bipotent state, you can expand them long-term, pretty much like this, as Hans said, like this oval cell that is activated during regenerative response. But in vitro, we don't allow them to differentiate unless we push them by changing the media, removing most of the growth factors, and adding notch inhibition to prevent the ductal fate, and then yet the cells can differentiate from that state ductal cells into hepatocytes. So and this you can do it clonally, so indeed this acquire a bipotent state in vitro, pretty much like the bipotent hepatoblast. In the liver, the only cell that is bipotent is the hepatoblast. And the ductal cells upon regeneration in particular settings. And the organoids kind of recapitulate that state in vitro. We showed that actually you not only can start from a mouse healthy liver tissue, but you get a healthy human biopsy, and the liver is very nice for that in a sense, because this is a donor, someone that dies and donates the organ, and then you can take a very little tiny piece of this donated organ and put it in culture in the same media condition, but with the addition of two factors, TGF-beta inhibition and cyclic IMP activation, and then the cells go on to grow for in the same, very similar rate for at least 6 months definitively. They retain the capacity to go from this progenitor state into a differentiated state where they can start acquiring a lot of hepatic markers, and also they are capable of having a functional activity. For instance, cytochrome activity is one of the detoxifying enzymes that metabolizes drugs in the liver. We also showed that they can produce a bit of bile acid, but now Hans's hepatocyte organoids show that the differentiation capacity of the hepatocyte organoids is much better towards the hepatocyte fate. And actually this, going from this ductal fate to this hepatocyte-like fate, it requires a lot of, it requires a 15-day protocol of changing the media in order to make them differentiate. What we found very interesting as well is that, here I'm just showing you a karyotype, but actually they retain the genetic stability over time after long-term passages. And we not only did karyotyping, obviously, but we did whole genome sequencing that is very, the organoids are very useful for that, because you can start them clonally, and then you can expand a clonal population and ask the question how many mutations you have acquired in culture. Recently, using an approach that was very similar to Hans' lab for the hepatocyte organoids, but modifying it slightly a bit, we had already data showing that the famous LGR5 is indeed a hepatoblast marker, and is a marker of the hepatoblast of the top of the hierarchy, and is a bipotent hepatoblast, and it only exists as a marker at very early time point in development when the liver bath is really forming at E90 10.5, and it will lose expression at E13.5. You almost lost the entire expression. But yet, if you take these E10.5 LGR5 positive cells in a clonal manner, you can grow them as hepatocyte organoids. But if you add hepatocyte media, if you change the same cells, if you put the same LGR5 positive cells in a cholangiocyte media, now you get cholangiocyte organoids that look very similar to the adult ones, single-layer epithelium of ductal cells. So what I want to explain to you today is how we've used organoid technology to model liver cancer, and maybe how we are exploiting it to understand cellular plasticity, in particularly in human liver cancer. So what we wanted to do is to establish an in vitro model that would recapitulate the different aspects of liver cancer. And liver cancer can be is a range of, in a sense, it's a range of diseases within a disease. You have hepatocellular carcinoma, and you have cholangiocarcinoma at the other extreme. The cholangiocarcinoma is close to a pancreas cancer than to a hepatocellular carcinoma itself. And then there is a mixed subtype, and we wanted to see whether we could use this technology to reproduce the different subtypes and retain the patient's specific characteristics. So Laura in the lab took this approach, and then we collected material from patients of hepatocellular carcinoma, mixed subtype, and cholangiocarcinoma, and and then we managed to expand them long-term in culture, and actually we saw that they grow as like a cystic structure, but like contrary to the healthy organoids, they grow as a cystic structure. They grow like a solid structure that it gets filled of cells. And I'll be happy to go into the questions if you are interested. But for the sake of time, just to show you that if you compare the transcriptome of the organoids to the transcriptome of the original tissue the organoids were derived from, you actually see a very beautiful correlation, not only in terms of the subtype, but also in terms of the specific patient. So in square is marked the strongest correlation. And the most interesting of it is that they are growing in the same media conditions, and yet, despite they are in the same, seeing the same signaling and the same factors, they kind of retain the transcriptome of the origin. And when they are hepatocellular carcinomas, and they have hepatocellular carcinoma markers, like AFP, and when they are cholangiocarcinomas, they have cholangiocarcinoma markers, for instance, like EPCAM. We also showed that they retain their genetic stability over time when we compared the mutations that the cultures have to the mutations of the patient, we see that there is an 84% concordance. And from this concordance, there is still some mutations around 20% we don't capture in the culture. We don't understand that. It might be selection, it might be probably selection. But what we find interesting is that there is no de novo mutations. So here we compared organoids or tumor organoids from an early and late passage after 4 months in culture or 5 months in culture to the original tissue, and actually at least they do not acquire de novo mutations. We found interesting that point. But to me, more interesting is that actually they retain the histological characteristics of the original tumor when we transplant them. This is something that, to my knowledge, cell lines cannot do, and something that organoids that retain the histological architecture, they do very well. So these were organoids that were from a tumor, from that particular tumor, that were transplanted after grown for 5 months in culture. They were transplanted into a mouse, and actually you can see that they retain the memory of doing even this desmoplasia, which is classical of a in that particular case is a cholangiocarcinoma. Together with Matthew Garnett and Haley Francis, Matthew is here, we actually asked the question whether we can use them for drug screening, and we showed that it is possible. You can plate them by a robot. A robot can plate them in these 384 well plates and put all these drugs, and actually we validated all of them, and we only found one difference between one of the lines and one hit on the screen. And as we expected, some are sensitive, some are resistant. There is some lines that are resistant to everything, which is quite scary. But what we found is that actually there is one in what we had an interesting hit, which is this ERK inhibitor, which was postulated by Jessica Zugman in a Nature Genetics paper where she mentioned ERK. So liver cancer is devoid of ERK mutations, so ERK pathway might be important or interesting. Nobody had tested it, and actually was a nice hit on our screen. Some were sensitive, some were resistant. We took some of the sensitive and some of the resistant, and here I'm showing you on the sensitive ones, and we showed that when we treat the tumor, when it's a real tumor, 100, you can really visually see it, and you treat it, then actually the tumor shrinks, and actually what we saw is that these are all tunnel-positive cells, while the vehicle grows, as we know, as in this glandular epithelium. And with that, I would like to thank the people in the lab, especially the people that contributed to the tumor project, which was Laura, together with other people in the lab like Robert and Patricia. And I have to acknowledge my funding, and I'll be happy to answer your questions. Thanks.